This presentation covers some supplemental materials over the cells. Cells are the structural and functional unit of life, and while this material is not located in any particular chapter in your textbook, I think that it's very important that you understand the nature of cells before we proceed in talking about different types of microorganisms. So, a little bit about cells. Cells are the building blocks of all living things. If it is alive, it is cellular in nature. That is because the cell provides a place for things to happen. There's an inside and an outside, and there can be a difference between those two spaces, which allows cells to carry out all the chemical activities needed to sustain life. All living things are composed of cells, and all cells come from pre-existing cells. Now, we talked before about the idea of spontaneous generation. We came to understand that life came from other life. So cells come from pre-existing cells. They don't just spontaneously appear out of nowhere. This is the essence of the cell theory. That is to say, cells are the building blocks of all living things. All living things are composed of cells, and all cells come from pre-existing cells. Now, in our studies, we're going to look at a couple of different cell types. These are two very broad categories. We have prokaryotic cells, which do not have a nucleus or many other organelles. And they are very small in size because having the organelles and the nucleus are necessary to be efficient enough to be larger. Prokaryotic cells are not the most efficient, although they are enormously numerous. Bacteria are prokaryotic cells. Now, eukaryotic cells contain a nucleus and other organelles. They are larger in size and more complex. Because they have those other organelles, they are able to be much more efficient in energy production, in function, and that allows them to be larger. Now, anything that is multicellular, humans, for instance, are made of eukaryotic cells. Let's look at the anatomy of cells. What we have here is a eukaryotic cell. It has a nucleus, cytoplasm, and a plasma membrane. The difference between that and the prokaryotes, like we said, is that prokaryotes do not have a nucleus, but prokaryotes still have cytoplasm and a plasma membrane. Now, not all cells are the same. We have four major groupings of cell types in our bodies, which can be divided down even further into something like 200 different cell types that our bodies use to function. So you get the idea that we, depending on the specialization of a particular cell, it may have more or less of some of the structures that we're going to talk about in a few minutes. Let's look at the nucleus. That is the control center of the cell, and it contains genetic material in the form of DNA. All living organisms have DNA as their genetic material. There are certain viruses that are RNA viruses. We talk about viruses in a separate category because they are really non-living in nature. So the genetic material is DNA, both in prokaryotes and eukaryotes. The nucleus itself surrounds the DNA in eukaryotes, and it is made of a nuclear membrane, which is very similar to the cell membrane. Within the nuclear membrane, there are a structure called the nucleolus, and there is sort of this amorphous goop that's known as chromatin. Now, the nuclear membrane itself is the barrier to the nucleus. And it's made of a phospholipid bilayer, just like the plasma membrane. It has a number of pores that will allow the nucleus to communicate with the outside. So materials can get into the nucleus and out of the nucleus, but it limits what can go in and out so that it protects the DNA that's within. Now, nucleoli are areas within the nucleus where there is a lot of activity going on. Specifically, what's going on is ribosome production. So depending on how much protein the cells make, it may need more ribosomes. So you can have one or two or more nucleoli. But the ribosomes are made within the nucleus, and then they migrate out of those nuclear pores into the cytoplasm where they are able to begin manufacturing proteins. The chromatin is unwound DNA. Now, we're familiar with DNA perhaps in the form of chromosomes, and we see those little X's and Y's in images all the time, but that's not the normal state for DNA. That's kind of like a wound up package, a skein or a ball of yarn. But normally, the DNA is unwound and just scattered throughout the nucleus, and that's what we see with the chromatin. There are proteins there that function to hold the chromatin together, that the chromatin or the DNA can wrap around. The plasma membrane outside of the cell is the barrier to cell contents. And again, it separates the inside from the outside and allows certain things in, certain things out. How that makes a difference relates to its permeability. But it is a double phospholipid bilayer in a fluid mosaic. And again, phospholipids have those hydrophilic heads that like water and the hydrophobic tails. So these phospholipids line up so that the lipids are back to back and the phosphors are out, exposed to the fluid that's either inside the cell or fluid that's outside the cell. Now, the phospholipids only make up about 60% of the cell structure. The remaining 40% is made of protein, cholesterol, glycoproteins, and actually glycolipids as well. These proteins have a number of purposes. The cholesterol serves largely to stiffen the cell, and glycoproteins either act as an adhesion factor or an identifying factor on cells. This is a graphic that illustrates the basic structure of a plasma membrane. And what we have is this fluid mosaic of phospholipids, and then we have these proteins that float and move through the membrane. Some proteins act as attachment points for structures inside the cell. Some proteins, such as this, act as a gateway for materials to pass through. You can see interspersed throughout cholesterol, which again acts as a stiffening agent, and then you see glycoproteins and glycolipids. These are of particular importance to eukaryotic cells. In mammalian organisms, it allows the body to identify what belongs to that body. So it allows the immune system to identify what might be a foreign invader. Now, between the nucleus and the plasma membrane, we have cytoplasm. It consists largely of cytosol. That is a fluid that suspends other elements. So we have, glucose, we have minerals, we have amino acids, we have a wide variety of things in the cytosol that the cell can use. Nonetheless, the cytosol is about 90% water. And then we have organelles. We have organelles in eukaryotic cells. We have one particular organelle that's in prokaryotic cells, which is the ribosome. The other organelles don't exist in prokaryotic cells. But organelles are the metabolic machinery of the cell, which allow them to function more efficiently and to produce products better, and as a result of that efficiency, allows them to be larger. Some cells also have inclusion bodies, and while it states here that these are non-functioning units, they do serve a purpose. They are just not an active part of metabolism. The inclusion bodies are storage areas, either for fats or starches or water or any of a variety of things, but things are stored in these inclusion bodies. So I think of these as kind of like the basement or the attic of a house. They're a spare storage space somewhere within the cell. This graphic shows you sort of a generalized cell. And you can see a variety of the organelles. You see the nucleus with its nuclear envelope. You see an organelle called the rough endoplasmic reticulum. You see smooth endoplasmic reticulum. There are ribosomes that exist in the endoplasmic reticulum, and out in the cell itself we have mitochondria, we have Golgi apparatus, there are a number of secretory vesicles. The cell membrane may have projections coming off of it, such as microvilli or flagella, and some cell membranes are very smooth. So this is, as I said, a sort of generalized cell. You'll never see one that looks exactly like this. So let's look at these cytoplasmic organelles. Ribosomes are very important. They are the site of protein production. We mentioned that earlier when we looked at the genetics component of this unit. Ribosomes are made of protein and RNA. They are the site of protein synthesis. Now, the ribosomes in prokaryotes are a little bit different than eukaryotes, but they still serve the same purpose. Ribosomes can be found in two locations. They are floating free in the cytoplasm, or they are attached to endoplasmic reticulum. Now, ribosomes that are floating in the cytoplasm are making proteins for the use of the cell itself. Ribosomes that are attached to endoplasmic reticulum or rough endoplasmic reticulum are making proteins that are exported. Other cytoplasmic organelles include the endoplasmic reticulum. These are fluid-filled tubes that are made, again, of a phospholipid bilayer, and they're there for carrying substances, packaging substances, storage of substances. And there are two types of endoplasmic reticulum. There is the rough endoplasmic reticulum, which is studied with ribosomes, and here we are building materials that may be used in the cell membrane or exported out. The smooth endoplasmic reticulum has a number of enzymes embedded in it which can function in cholesterol synthesis, breakdown of materials, fat metabolism, detoxification of drugs, and again a pathway for materials to be carried throughout the cell. An area of processing is the Golgi apparatus. It modifies and packages proteins, produces several different types of packages actually, including secretory vesicles, cell membrane components, and lysosomes. Now, this is a good place to stop. We're going to pick up and continue with the Golgi apparatus in part 2 of this presentation.